In this video, we will discuss about the Sialyl Lewis glycoform and the pseudopod protrusion. So the Sialyl Lewis is the structure that is shown on the left hand side, and they are usually found to be overexpressed in the cancer cells, especially in the pancreatic cancer cells. These are also known as truncated O-glycan structures. So, and there are different glycoforms of the Sialyl Lewis are found. For example, this one is the extended core 1 structure, and it can also be found in this core 2 structure form that is shown here. And there are many reports, there are many research that also reported this biantennary that contains both the extended core 1 and core 2 structures that is shown here. And there is antibodies, that is MECA79, and these antibodies can bind to these epitopes that is highlighted. However, these epitopes, it can bind to these epitopes only when it is available in the form of biantennary. It will not bind to this epitope directly in this extended core 1 structures or the core 2 structures. So this is unique property of this MICA79 epitope, and this binding of these epitopes will also cause activations of this GTPase that is important in the pseudopod protrusion. So what is before we going to this pseudopod protrusion, what is this pseudopod protrusion? Pseudopod protrusion is mainly occurs when the cells is trying to move from one place to another state, or we can say that when cells are in mesochemical state, how the mechanical properties changes within the cells is mainly dominated or explained by this pseudopod protrusion. And here it is explained through with the help of this L-selectin and the monocytes interactions. Monocyte contains, apart from here in the previous video we have discussed ERM, that is Ezrin-Radixin and Ezrin-Radixin and also Moesin. However, in the case of monocyte, the radixins are not expressed that much, whereas Ezrin and Moesins are expressed in higher amount. So when they interact with this L-selectin, so and from this Ezrin interactions, it will also cause the activations of this P13K. At the same time, the interactions of this Ezrin to L-selectin, that is to this FERM region, it will cause the phosphorylation of this FERM region that helps in the binding of this SYK. This SYK helps in the phosphorylation of this VAV, and this VAV in the presence of this PIP3. So how these PIP3s are formed? P13K helps the conversion of this PIP2 to PIP3. PIP3s are dominated here, mentioned here as a red dot, and the PIP2s are mentioned here as a purple dot. So this PIP3, when interacted with VAV, that is shown here, it helps in this RAC GTP. One more thing, that P13K, when the phosphorylations of this VAV occurs, it also affects this GTPase that is shown here. That VAV, phosphorylated VAV interact with this RAC GTPase, and this helps this actin remodeling or change in the physical properties within the cells, and that is shown here, that how these interactions, that is Ezrin to this L-selectin, cause the intravasculations of the cells or extravasculations of the cells. And this is basically one of the properties or one of the phenomena, one of the process that pancreatic cancers usually goes through when they are migrating from pancreas to secondary organ. We will discuss how these things occurs in the form of loop in our subsequent videos. Here I have only covered panel A of this figure 4. We will discuss panel B and panel C in the next videos. I hope you understood some basic idea about this pseudopod protrusion. If you need to understand more about this in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.